Okay, hello, welcome all to the 11th Journal Club meeting. To get your points, scan the QR code and then use the word insulin. So the purpose of Journal Club is just to introduce you to published literature. It gives you exposure to research papers in a low stress environment versus working in like a lab or at whatever your graduate school is. It also provides a space to practice presenting research, and it exposes you to different areas of academia. So the expectations for participation is to have read the article beforehand, to bring up own ideas, question the methodology of the paper, discuss the applications, practice reading literature, identify common structures to the research papers, and take notes and annotate if needed. So this week we're going to be covering the in vivo reprogramming of adult pancreatic exocrine cells to beta cells. It's actually a paper from 2008, so it's a little older, but it's a paper that led to a lot of different therapeutics that are going to be coming on the market relatively soon for type 1 diabetes. So a little background on the topic, exocrine cells in the pancreas, they just secrete the digestive enzymes like protease, lipase, amylase, things that break down your food once they're in the intestine. And then the endocrine cells, they're arranged in islets. They secrete hormones, including insulin, glucagon, somatostatin. So beta cells are going to produce the insulin, and then alpha cells produce glucagon. Type 1 diabetes is a chronic autoimmune disease where the beta cells in the islets of the pancreas are destroyed, typically through an immune response, but it always results in insulin deficiency. The typical diagnosis, besides to differentiate between type 1 and type 2 diabetes, is to see the presence of serum autoantibodies, the antibodies that your body secretes that attack your own B cells. And then pluripotent cells, they're stem cells that have the ability to undergo self-renewal and give rise to all the tissues of the body. So the purpose of this experiment was to identify and test the specific combination of transcription factors that reprograms differentiated exocrine cells into resembling cells that resemble beta cells. So the methods into design, to determine the transcription factors, they use an in situ hybridization screen for over 1,000 transcription factors to identify the cell type specific expression in embryonic pancreas. This was just to determine what transcription factors would need it to become the endocrine cells. And then the induction of the insulin plus cells in adult mice was done by using an adenovirus co-expressing each transcription factor with nuclear GFP. I'll throw a question out there. Why would they use an adenovirus? It's a little more complex, but. What is an adenovirus? It's just a class of viruses. The reason they actually use those a lot of times in getting to co-express is because they're double-stranded DNA. So it'll actually insert into your genome, which causes the expression to change. That's why once you have herpes, like you have it for the rest of your life, because it's actually like the virus inserts itself into your DNA. So you'll often see it anytime they want to change the transcription factors or the expression of cells. And then a lineage analysis was done to determine the origin of the insulin plus cells to see if they were actually the new like induced cells or just the original beta cells. And then to determine if the cells resembled beta cells, they looked at the morphology, so the size and shape. They use immunoelectron microscopy and then molecular marker analysis to determine how closely those induced beta cells resembled the true beta cells. And then the analysis of insulin release, they did this by inducing diabetic mice and then conducting glucose tolerance tests to see how well those new cells functioned. And then to determine if the presence of the inducing factors were still there, they did a reverse transcriptase PCR to detect the presence 1 month and 2 months following the induction. Okay, so for the results. So for the first part, like we talked about, the combination of the 3 transcription factors to induce, as you can see, looking at the presence of the GFP plus cells, it required all 3 NGN3, PDX1, and MAFA to get the higher levels of expression. And then as you can see, the adenovirus preferentially infects the pancreatic exocrine cells. So the exocrine cells are going to be these out here, while the islet endocrine cells are going to be here. And then as you can see, lighting up shows that these exocrine cells were actually induced. Okay. And then to determine if the new beta cells originated from differentiated exocrine cells, they looked at the GFP viral infection 10 days. So most of the amylase plus cells, which is amylase comes from the exocrine cells, they come from the mature exocrine cells. But then after infection, many of the beta gal and insulin are present from those differentiated cells. Okay, so then the endogenous and induced beta cells are indistinguishable from the morphology and ultrastructure. So this is basically determining if those cells that are induced resemble the isolate beta cells. So as you can see, they're similar in size and structure. The exocrine cells are going to be much more narrow than these islet cells, but as you can see, the induced beta cells begin to take on that more circular shape, and then more similar size as well. And then the endogenous and induced beta cells contain small insulin granules, distinguishing them as endocrine cells, and lack the zymogen granules, which would be seen in exocrine cells. And then the molecular markers of induced beta cells 1 month after the co-expression. So 4A, as you can see, shows the glucose transporter 2, which would be characteristic of those endocrine cells, glucokinase, and then prohormone convertase. And then these 3 here show the different transcription factors that are characteristic of beta cells. And then finally, the ones down here show that the induced beta cells don't express amylase, glucagon, or somatostatin. So amylase showing that it's no longer functioning as an exocrine cell, and then these 2 showing that it functions as beta cells versus other endocrine type cells. Additionally, the induced beta cells remodeled vasculature and amylase hyperglycemia. So what does that mean? So basically, they induce angiogenic remodeling. Basically, that just means bringing the blood vessels closer to the cell so it can get the new blood supply. Additionally, there's improved blood fasting glucose levels in diabetic mice after the injection compared to controls. So right here, so this is going to be the non-diabetic control. This is going to be the diabetic control, and then this is going to be the induced cells. So as you can see, the blood glucose levels dramatically drop in comparison, showing that most of the activity of the insulin is recovered. Additionally, it tested the serum insulin and then the average beta cell numbers after injection. So as you can see, wild type, no diabetes, the insulin functions normally, and then these 2 show following injection. Okay. So what does all this mean? So it shows that fully differentiated endocrine cells can be directly reprogrammed into cells that closely resemble beta cells. This means that they don't have to de-differentiate and go back to the stem cells that they originally came from. And this is achieved using a combination of 3 reprogramming transcription factors. So the NGN3, PDX1, and MAFA, they were all shown that all 3 are necessary to induce the cells. Additionally, the induced beta cells remain as single cells and don't reorganize into isolate structures. The reprogramming of exocrine cells to beta cells occurs relatively fast, with the first insulin producing cells appearing after 3 days. So what are the limitations? So some of these are limitations of the study itself, others are limitations of its application. So the first one, induced beta cells don't organize into isolate structures and lack the organization, and their lack of organization of induced beta cells impairs their function. So the isolates are typically drawn together because that way they can bring the blood closer, and then they all function within a same location. Additionally, there was a lack of long-term follow-up of the beta-induced cells and function in the insulin production, so they only checked up till 2 months, so there wasn't any further progress to see if they continued expressing beta cells. Additionally, there was a small number of animals studied, so many of the findings were only found in 3 of the mice. So it would have been better to prove statistical significance if they had a higher number of mice studied. Additionally, the PCR was performed on populations of cells instead of the single cell resolution immunohistochemistry. This shows that it's unclear how many cells actually express the markers or at what levels. And then what do you think some of the strengths of the study would be?
As well, they didn't do it for a longer term. They didn't do it up to a month. So taking those measurements within that time frame is probably more interesting. Seeing how the time of the induced cells affected the insulin production of the rats.
Yeah, I would agree. Additionally, I would say that they did track the origin of the cells to determine if they were actually differentiated from exocrine cells versus just expanding endocrine cells. So this study actually has a ton of super cool and future studies. There's a lot of follow-up with this. There's drugs that are in clinical testing right now and hopefully will be on the market soon. But there's several things in the paper that need to be understood better. So studying why the particular 3 combination of transcription factors are sufficient for reprogramming. That mechanism still isn't known. It was only observed. Additionally, study for the strategies that promote the aggregation of induced beta cells to reduce the full glucose responsiveness. So as you saw in the previous, there wasn't a full production towards more similar to the wild type. A lot of this would have to be due to the fact that they still remain segregated like the exocrine cells. And then additionally, the suitable culture conditions for in vitro generation of beta cells is unknown. Yeah. Any other ideas what of this potentially being used for? Yeah, basically there's been a lot of cool studies that have come from this paper, potentially trying to cure diabetes in humans. They've developed a similar induction pattern in humans that's actually going through clinical testing right now. Let's see. Yep, so this clinical outlook, so there's a possibility of generating patient-specific stem lines for therapies. This could be many different types of autoimmune diseases or diseases in general, genetic diseases as well. as long as you're able to affect the expression of the cell, you're able to get the preferential treatment. Additionally, the major outcome of this would be a treatment of type 1 diabetes through the induction of exocrine cells to beta cells. Additionally, this could be used for treatment of tissue repair and regeneration. It's seen that beta cells destroyed by various different means, you can induce other cells to become those cells. Any other ideas of what could this mechanism or anything from this paper could potentially be used in the future?
Yeah, like more generally, just the use of transcription factors to alter genes.
Yeah.
They use like further than insulin. And if the further research of this actually works, then it's on different things too, other diseases like mentioned.
Yep. Yep. And then references, and then any questions?
So, yeah, so you said that they don't know like the exact mechanism of why those 3 transcription factors work. So how did they pick those 3 transcription factors if they didn't really know that they were gonna work?
So they basically looked at like which transcription factors are expressed in different cells, and then they did that like 1,000 different combinations of transcription factors. So they narrowed down which individual transcription factors induced the expression, and then tried different combinations of those. And from that combination, they saw that that one works the best.
Kind of like a trial and error type.
Yep.
Right.
Yeah. And I mean, there could be another transcription factor involved in the process. They don't know that. If it's one that they hadn't tried, that's also a possibility as well. Yeah. Any other questions? Okay, I'm gonna end the recording then. Thank you.